Hello students, today I'll be teaching to you all the next chapter, that is biotechnology and its applications. So you people have already studied the chapter biotechnology, principles and processes, where you have studied the different technologies that are involved in the biotechnology, which makes use of live organisms to produce the products and services. The technology is involved along with the microorganisms, so the living system is involved. Together they combine to produce the products. Now the products are used by the humans, for various applications, right? So we have seen different processes as well as different techniques of the biotechnology, such as PCR reaction, agarose-gel electrophoresis. Each technique has once again its own importance, right? But what is the ultimate goal of the biotechnology? We have seen what are the processes involved in the biotechnology, right? But why we should learn these principles and processes? What is the need to learn these principles and processes? The need is that the ultimate goal of biotechnology is to produce the products in large scale. We have seen the traditional biotechnology which just makes use of live organisms to produce the products, whereas the modern biotechnology deals with the manipulation of these live organisms. Either you can insert a new gene in it, you can modify its genetic composition to produce a desired product, right? So moving on to the next level, that is we are going to produce into the large scale, wherein that is applicable for various streams. The products that are obtained from the biotechnology, that is from the modern biotechnology, that is utilized for various fields, right? So biotechnology has got wide range of applications in various fields. It is used to produce the biopharmaceuticals and biologicals. So what are these biologicals? Like there are certain therapeutics that have been extracted from the live organisms, such as vaccines. Either they are produced in these live organisms or they are extracted from these live organisms. They are known as biologicals, right? So the biotechnology is going to produce these biologicals or the biopharmaceuticals in a large scale, that too in a controlled manner, right? In this growing population, the demand for the therapeutics is very high. To reach or to fulfill the demand, the products are being now produced using the biotechnology processes. That is using recombinant DNA technology. Large amount of products can be produced in lesser time. So let us see some of the applications of the biotechnology, right? First application, if you see the first application, therapeutics. Therapeutics means production of drugs in large scale using recombinant DNA technology or the biotechnology processes. Large scale production of drugs to treat various diseases involves therapeutics. And not only producing the drugs, as well as using the biotechnology techniques, their effect on the disease or their effect on the patient can be studied, right? So how the biotechnology plays a role in therapeutics is it helps in the large scale production of the drugs. For example, human insulin. So insulin is being used to treat diabetes. Usually in the body, the pancreatic cells produce insulin to regulate the sugar levels, right? In case the insulin is produced less in amount or it is not produced at all because of the damaged cells or the body is unable to utilize the insulin, then the patient has to take insulin externally in order to regulate the body blood glucose level. How this external insulin is produced? In the body, it is produced in the very less concentration, and therefore, so in order to fulfill the need for various patients, the insulin requirement, the drug has to be produced in large quantities, in a large scale. So this is achieved by recombinant DNA technology. The insulin was produced from the microorganisms, that is bacteria. Then it is extracted from these cells, purified using downstream processing. Finally, it is packaged and distributed to the end user, where the diabetic patients use the insulin. So therefore, biotechnology has applications in therapeutics. The therapeutics use the biotechnology processes to produce large scale production of drugs, as well as biotechnology techniques are used to study the effects of the drugs on the human beings, how the effect is going to stay, whether the treatment is effective or no. All these things can be studied using the biotech techniques. Another application of biotechnology is diagnostics. For any disease, in order to start the treatment, first thing is the diagnosis has to take place. The disease has to be diagnosed in order to find the root cause of the disease. Then only the proper treatment can be given. Right? So, in the diagnosis also, the biotechnology products such as the proteins or enzymes that are obtained using recombinant DNA technology are used to diagnose various diseases. For example, AIDS, HIV, right? AIDS is once again, it is being diagnosed using the recombinant DNA technology techniques. Or if there is any defective gene present in the body that is leading to the malfunctioning of the biological processes, that defective gene can be identified using the biotechnology techniques. So therefore, the biotechnology has got its application in diagnostics to diagnose various diseases. How the products or such as enzymes or proteins or genetically engineered organisms, these are going to be used in the diagnosis of the diseases. They are used in manufacturing certain test kits such as ELISA. It is enzyme linked immunosorbent assay. This kit, it is based upon the antigen and antibody interaction. So whatever the antibodies required are being produced for this kit through biotechnology, that is through recombinant DNA technology. Not only for ELISA kit, there are certain monoclonal antibodies are produced. These are the antibodies that are being extracted from same immune cells, which are going to act against a single epitope of the antigen. Right? In an antigen, the protein part which is going to elicit the immune response, that is known as epitope, and this epitope is recognized and the antibodies are produced. These antibodies are going to act against this epitope. These are once again produced using recombinant DNA technology. Those antibodies are known as monoclonal antibodies. So therefore, in diagnosis, the enzymes and proteins produced are used for diagnosis purpose, or the techniques of the biotechnology such as PCR reaction, they are used in diagnosing the diseases. Another application of biotechnology is in production of GMOs. What are these GMOs? GMOs are genetically modified organisms. GMOs are genetically modified organisms. These are also known as transgenic plants in case of plants, or in case of animals they are known as transgenic animals. Plants whose DNA is modified or altered using genetic engineering techniques, they are known as genetically modified plants. Means the genetic composition is being varied. The genetic composition is designed in these plants either by removing certain genes or by adding certain genes, that is known as genetically modified crops in case of plants. Now these crops are being manipulated for agricultural purpose. For agricultural purpose, to create the plants that are pest resistant, that are drought resistant, or in order to produce the high yields, the plants are modified. So therefore, these modifications once again they are based upon the recombinant DNA technology. The pest resistant plants are produced, so therefore the artificial or the fertilizers, the chemical based fertilizers usage will be very less, because the plant itself has the resistance towards the pest. So like this, the agriculture is also having biotechnology application. Next, another application of biotechnology is bioremediation. It is bioremediation. What do you mean by bioremediation? Bioremediation is usage of modified organisms in order to treat the contaminated media, water, soil, or any subsurface material is known as bioremediation. Processing of the waste, processing of the contaminated media, processing of the contaminated water, or processing of the subsurface material using the organisms is known as bioremediation. In bioremediation, in a reactor vessel, the controlled conditions are maintained, a controlled pH, temperature, what are the nutrients required, everything is maintained for the growth of the microorganisms to act upon this contaminated media. So you are going to treat this contaminated media in a controlled manner using the conditions, using the microorganisms in order to treat the contamination. For this purpose, how the biotechnology is contributing is, it is going to provide genetically modified organisms. Right? It is going to provide genetically modified organisms. These organisms are going to act upon this contaminated media and remove the contamination. Not only it is going to provide the genetically modified organisms, it is also going to provide in maintaining the conditions. The biotechnology is going to maintain the conditions. The techniques of biotechnology helps in maintaining the proper pH. It is going to maintain the proper temperature. What are the nutrient concentration required? Everything is maintained by the biotechnology processes in bioremediation process. So, biotechnology has application in therapeutics, in diagnostics, in GMOs, especially in producing genetically modified crops for agriculture purpose. Also, it is going to have application in bioremediation to treat the contaminated media using the microorganisms under controlled conditions. That is, in a controlled conditions, which is going to treat the contamination by altering the environmental conditions that stimulates the growth of the microorganisms which degrades the targeted pollutants. Now, here the pollutants could be oil, petroleum products, solvents, pesticides, which are not so easily degradable, right? They require the microbial action, and this microbial action is provided by the genetically modified organisms. There are certain organisms which consist of the genes called degradative genes. This encodes proteins required for degradation of pollutants. So, the genetically modified organisms will have the capacity to degrade oil and petroleum hydrocarbons, which is very difficult to clear the contamination. So the genetically modified organisms helps in degrading oil and petroleum hydrocarbons, right? So the biotechnology provides genetically modified organisms that helps in degrading the target pollutants, as well as it is going to maintain the conditions that stimulate the growth of these microorganisms. Another important application in biotechnology is waste treatment. Important application of biotechnology is waste treatment. So the waste treatment involves the activated sludge, which is the treating of the sewage and wastewater in an aerobic environment using the microbes. Activated sludge is treating the sewage and wastewater in an aerobic condition using the microbes. In this, the pathogenic microbes are being removed, as well as it also involves biosorption, which means the microbes are used to remove the metal species from aqueous solution. Biosorption involves removal of the metal species using the microbes in the solvents. Then once again the microbes that is going to act upon the metal species are produced by recombinant DNA technology or genetic engineering. Clear? So, usage of modified organisms or enzymes in producing the metal-free solvents, that is known as biosorption. Another important application of biotechnology is energy production. Energy production. So, usage of the modified organisms or enzymes to produce fuels. The microorganisms are used to produce fuels required. The fuels are required for running the vehicles or for running the missions in the industries. The fuels are required. How these fuels can be produced using the biotechnology? That is, once again using the modified organisms. Now these modified organisms are going to undergo fermentation process and they are going to produce the products. The fermentation process is what? Action of the microbes on the sugars and they are going to produce the alcohol. So, when the starch is acted upon by the microorganisms, it is going to produce ethanol. Fermentation of starch produces ethanol, which is used as alternate source for petrol in road transport in the foreign countries. In some of the countries, this bioethanol is used as a alternate source for petrol and it is used in road transport. Not only bioethanol, there is the biodiesel. Biodiesel is also produced once again using the oil crops. Using the oil crops such as soybean, the biodiesel is produced using the recombinant DNA technology. So, these are the applications of biotechnology. They are used in therapeutics, they are used in diagnostics, they are used in the production of GMOs for agriculture, they are used in bioremediation to treat the contamination using the genetically modified organisms, as well as the biotechnology techniques maintains the conditions for the growth of the organisms for bioremediation, as well as in the waste treatment. The biotechnology is going to produce the modified organisms that is going to treat the metal species in the solvents, as well as the biotechnology has application in energy production, that is by the fermentation process using the modified organisms, biofuels can be produced. These biofuels are used as a fuel for road transport system in some of the countries, such as bioethanol and biodiesel. So these are the applications of biotechnology. So now let us see what are the three critical research areas of biotechnology. What are the three critical research areas of biotechnology? Why the research of the biotechnology is focused on what? For what the biotechnology research is going to take place, right? For example, now in the current scenario, if we see, because of the COVID-19, there is a large investment on the research centers to produce a vaccine for the coronavirus, right? To produce a vaccine for the coronavirus. So that is a research area. Once again, this is based on the biotechnology only, right? How the vaccine they are going to produce, and if you see the upstream processing, downstream processing. Now the vaccine, if it is produced in the R&D level, it is not sufficient. It has to be produced in the large scale to reach the patients, right? So now you might have understood why we require drugs in large scale, right? Because once the covaxin is being produced, that is now still the research is going on, but if it is commercialized, then it has to be supplied to the whole India. The three critical research areas of biotechnology is providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. See the important research area. Now if you see the case of COVID-19, here the research area is to produce a product or a vaccine, right? So it could be this could be in the form of a microbe, or it could be in the form of a pure enzyme. That is important, means either you want to produce an organism that produces a product that can be used as a drug, or directly you are going to produce the drugs in the organisms. So that is meant as a catalyst. So providing the best catalyst in the form of an improved organism. Improved means, see, in the human beings usually the insulin is produced, but we want to produce the insulin in the large quantities. Therefore, the gene coding for insulin is inserted in the E. coli cells. So, the E. coli cells or the bacterial cells are genetically modified. So, the genetically modified organisms is production of those organisms is one of the research area of biotechnology. So, either we are going to produce the modified organisms, or we are going to directly produce the products of the organisms, such as proteins, it could be, or any other product, enzyme, hormones, all these are the best catalyst. Means what are the research we are doing to produce the drugs or the organisms is one of the critical research area of biotechnology. Second, creating optimal conditions through engineering for a catalyst to act. So one of the example or the application we have seen in biotechnology, it was bioremediation. So what happens in bioremediation? The treatment of the contaminated water is going to take place in presence of microorganisms. For these microorganisms to act upon, it needs certain conditions, right? For any microorganism that has to act upon, it require certain conditions such as pH, temperature, oxygen requirement, it could be, right? It could be the temperature, all this, or the concentration of the substrate for the enzyme, or for an organism it could be concentration of the inducers to produce the product. All these are the optimal conditions. So based upon, once again, the biotechnology performs its research for providing the optimal conditions for the catalyst to work. Now the area of interest of the research in biotechnology is to produce a improved organism, a genetically modified organism. So for that organism, in order to act, in order to grow and to produce the desired product, it needs certain conditions, growth conditions. So these growth conditions are also a part of the research of the biotechnology. The biotechnology is going to produce the different optimal conditions for the growth of these organisms. Means the biotechnology research also includes the conditions, the optimum conditions, such as how the optimum conditions are studied. Once again, based on the research, the optimum pH for a particular organism, optimum temperature for the particular organism, optimum substrate concentration, optimum inducer concentration, what are the nutrients present in the growth media, everything is once again researched and it is been identified for the catalyst to work best, right? And other critical research area of biotechnology is downstream processing. It is downstream processing. What is this downstream processing? You people have studied. It is separation and purification of the product from the cells and recovering the product in its purified form is known as downstream processing, which involves the separation and purification techniques. Okay, once again, now what happens in the first research area? It is producing an improved organism. It is producing genetically modified organism, right? In the second research area, the conditions for the growth of this modified organism are being produced. In the third research area, now the product is produced from the organism. Next, it is to extract the product in its purest form. That is, once again, there should be studies on the separation and purification techniques, that is the downstream processing, to purify the protein or the organic compound so that it is going to give its maximum activity. If your product is pure, it will give you more activity. For example, gold. If you see the value of the pure gold, the 24 karat gold, its price is very high because it is the purest form. Similarly, when the product is produced within the organism, it will be associated with other molecules also. So, therefore, it has to be segregated, it has to be purified, and that purest form is preserved with the stabilizers, and then it is packaged and it is marketed. So this all is once again involved in downstream processing. So the research areas involves, one is to provide the best catalyst, the research they are going to do, and they are going to modify the organism. Second, this modified organism, it is going to have a set of conditions. So those conditions are created for the growth of this organism. that is providing the optimum temperature, pH, pressure, substrate concentration, inducer concentration. Then finding out the separation techniques to extract the product in its purest form. These three are the critical research areas of biotechnology. Right? Then we are going to see one by one application in detail. So we are going to see some of the examples. We have seen that biotechnology has application in providing the GMOs, by producing the GMOs, that is genetically modified organisms. So one example is GM crops, genetically modified crops. How this genetically modified crops is having advantage in agriculture. So we are going to see the application of biotechnology in agriculture. The application of biotechnology in agriculture. So let us see in the ancient days or few years ago how the food production enhancement was done. What were the ways to produce the high yielding food productions, right? The agriculture has undergone very much high development to produce increased food production. So what were the steps taken to enhance the food production? Okay, there was agrochemical based agriculture, there was organic agriculture, there was genetically engineered crop based agriculture. These three agriculture techniques have enhanced the food production. Why? Because of the growing population, the demand for the food is increasing. In order to meet the demand, there should be certain modern techniques involved to produce the increased yields in the food production. So, the different ways that were tried to enhance the food production, one such way is agrochemical based agriculture. So, what happens in the agrochemical based agriculture? That includes usage of fertilizers, insecticides, herbicides to increase the yield of crops. That involves fertilizers, chemical fertilizers, insecticides, herbicides, which is going to increase the yield of the crops to meet the increased need for the food. So that is one of the way to enhance the food production. That is agrochemical, usage of chemical fertilizers, pesticides to enhance the food production. Another way is organic agriculture. That is usage of fertilizers of organic origin, which are depleted from chemicals which are of organic origin, are used for agriculture, that is growing crops or farming that is based on usage of fertilizers of organic origin, that is called organic agriculture. For example, if you see organic fertilizers such as manure, feces of animals, or there could be bone meal, the animal bone powder, and the slaughter waste. What is the after killing the animals, whatever the waste that is produced, they are used as manures that are added to the soil to improve the growth of the crops. So that was again one of the way to enhance the food production, right? One is agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture is the another way to enhance food production. In case of agrochemical based agriculture, it includes usage of fertilizers and insecticides. In case of organic agriculture, it uses fertilizers of organic origin such as feces of animal or the animal bone powder or the slaughterhouse waste, which is going to produce certain nutrients for the growth of the organism. Another is genetically engineered crop based agriculture. This is usage of genetically modified plants or crops to get increased yield. No doubt using the agrochemical based agriculture, there was a growth in the food production that has led to the green revolution, right? The green revolution which has taken place between 1915 to 1960s means the measures were taken, the initiatives were taken for the increased agricultural production worldwide in developing countries to meet the high food demand for growing population, right? So whatever based upon the agrochemical based agriculture and certain modern facilities were being implemented to produce the high food production. Some of the steps taken in green revolution, that is high yielding varieties of seeds, hybrid seed varieties were used. High yielding varieties of seeds means which has the capacity to produce high yield crops. Those seeds were being used based upon the hybridization techniques. Alright, hybrid seed varieties were used to enhance the yield. Not only that, chemical fertilizers were used, agrochemicals were used, pesticides and herbicides were used to enhance the agricultural growth as well as improved irrigation facilities. That is once again very important to produce the high food production, and new methods of cultivations were implemented, right? Rotation crop based or what type of cultivation method, mixed crop, everything was being tried and which to increase the food production. No doubt with these techniques the food production was tripled and it has almost increased the food production to meet the growing population. But there are certain drawbacks of agrochemical based agriculture or organic based agriculture. So this couldn't continue further to keep constant the high food production. It didn't help further to continue the high food production. There were certain drawbacks. Now the green revolution, it was led in 1950 to 1960, which has almost tripled the food production by the usage of high yielding varieties, chemical fertilizers and agrochemicals. Okay, but still it was not satisfied because the population is increasing day by day. So usage of fertilizers and pesticides for improved crop varieties are expensive for farmers to afford, right? The artificial or the chemical fertilizers which were used for the producing high yielding varieties, okay, for the high yielding crops, it was very much expensive for the farmers to afford. That is one reason. Other reason is growing population was not fed enough with the current food production. The growing population was not enough fed with the current food production because the population is increasing day by day, and whatever the food production is taking place using these is not fulfilling the requirement. And in order to produce the increased food production with the high yielding varieties of crops, it requires large amount usage of the chemical fertilizers, and the usage of the chemical fertilizers for a longer period is going to produce harmful effects on the environment. The chemical fertilizers used produced harmful effects on the environment because the chemical traces were left out in the environment, which is going to pollute the environment and cause certain side effects in the organic life. In case of the living system, it is going to create certain side effects. Therefore, the facilities that were provided for the green revolution were no longer continued further for the enhanced food production. But There is another option, that is genetically engineered crop based agriculture by the usage of GMOs. That is, genetically modified organisms were used to produce the high yielding varieties, okay, to increase the yield of the crops. Clear? So next we will discuss about the some of the advantages of the genetically modified crops or plants in agriculture, and also we will see some of the examples of agriculture, that is examples of genetically modified plants in agriculture.